I'd like to introduce you to our speaker today, Dr. Laura Cox. She is Assistant Professor of Neurology at the Romney Center for Neurological Diseases at Harvard Medical School and Brigham and Women's Hospital. And Dr. Cox began her career in anaerobic bacteriology. She was trained and board certified as a medical technologist, and then worked for 2 years as a clinical microbiologist. She obtained her PhD at NYU in Dr. Martin Blazer's lab, where her work demonstrated the early life antibiotic treatment results in lasting changes in metabolism. Dr. Cox is now investigating the influence of intestinal microbiota in immunity and neurodegenerative diseases, including multiple sclerosis and Alzheimer's disease. In addition to funding from the Women's Alzheimer's Movement, Dr. Cox has received a fellowship from the Brigham Research Institute's Women's Brain Initiative for her studies of gender-dependent interactions between microbiota and diet in a model of Alzheimer's disease. Dr. Cox has been funded with 2 projects at Cure Alzheimer's Fund, and today is going to speak to us on the potential for harnessing the microbiome to prevent and treat Alzheimer's disease. And this is a version of the talk that she gave last November to the United States Senate at the request of Senator Ed Markey. With that, I would like to welcome Dr. Cox.
So as Kelly mentioned, I am a clinical microbiologist by training. I came at this from an angle of an infectious disease biologist. And then I really wanted to understand not just how, getting sick for a week can, you can bounce back from that, but how do these long-term chronic interactions with our microbiome really shape brain health? So I'll talk a little bit about that today. But first, I really want to talk to you all about, the impact that the Cure Alz Fund has really made in our research. So as a microbiologist, I joined the lab of Howard Weiner, who's a neuroimmunologist, back in 2015. And we started working on really studying the gut microbiota, how it influenced the immune system, and how it influenced the brain. And Cure Alz Fund was one of the very first organizations that really believed in our work and funded one of our projects. And based on that initial funding of what I guess at the time could have been seen as a high risk proposal, we were able to get solid preliminary data. And from that approach, the National Institutes of Health and other foundations, and really expand our entire research program. So this award came at an important time in my career when I was a postdoc, and was able to build this funding portfolio to support many new training scientists. This is now the established Cox Lab, which we started back in 2021. So I want to say that really these funds make a really big impact when we're trying to tackle new areas. And what I would love to talk to you about today, and bear with me, I'm just moving the Zoom control so you can see the slides. I'd like to talk to you about the gut microbiota, and we think about microbes in a negative context oftentimes. But what I want to tell you today is that they have many, many beneficial functions. So first, they help us aid in digestion. They produce vitamins. They can shape our immune system. They protect us from infections. They can actually stop you from getting diarrhea if you eat a bad potato salad. They can regulate our metabolism, and they can also influence the brain. And within your body, within your gut, you have about 100 trillion microbes. So all of you can pat yourselves on the back. You're supporting a community of 100 trillion today and every day. And so we want to think about how those microbes in our gut can affect the brain, such a distant organ that seems really separated. And so we can think about many different pathways. First, bacteria can secrete substances that make the cells in our gut secrete neurotransmitters. But secondly, bacteria can actually make all the neurotransmitters that we know about. And this can have a profound role in mood, anxiety, depression, and many other comorbidities that are often seen with Alzheimer's disease. We know that microbes can influence the immune cells in the gut, And those immune cells can then go into the brain and shape immune responses. And this could actually have a beneficial function. One thing that happens in Alzheimer's disease is there is a development of amyloid plaques, and immune cells can traffic into the brain, see those plaques as damage, chew them up, clear them out, and actually have a protective role. Microbes can also signal through the vagus nerve, so directly through our nervous system. But one of the things to remember, it's actually a two-way street. So if you've ever been feeling really nervous or anxious about something or really excited about something, your stomach feels kind of off, or the butterflies in the stomach feeling, it's because you're secreting neurotransmitters into your gut that will change your gut motility and it'll change your gut microbiota composition. So when we really think about Alzheimer's disease, let's think about the steps in the process and the pathology. We know it starts from this accumulation of A beta plaques and tau tangles. And this is really a problem from too much protein building up. So if we could find ways to clear that protein over the long-term course, those could be really helpful strategies to tackle AD. We know that aging and environmental factors play a major role. There's genes that have been identified in Alzheimer's disease, but largely it happens in the sporadic population. It's not really always assigned to genetic differences. We also know that there are age-related changes in the gut microbiome. And as we age, our gut microbiota ages, and there can be consequences of that. And then finally, again, the immune system in the brain, these cells that we call microglia, and then cells that come in from our blood, cells we call monocytes, can play a really important role in clearing plaques. But if they get overactivated, they can actually contribute to the disease and cause damage and loss of brain cells. And so we think that the gut microbiome can actually influence all of these processes, the beginning of the pathology, including making APP and tau tangles, by influencing the microglia and by influencing peripheral immune cells. And this is really at the heart of the project that Cure-All's Foundation funded since 2019. There are many different pathways by which the gut microbiota can affect the brain, but we also have to remember that we don't live in a bubble, and there's many things in our world that can shape the microbiome. Such as aging, diet, antibiotics, where you are in the world, what microbes you have in your environment, your lifestyle, and also biologic sex. All of these factors can push or shape the gut microbiome, which might affect the immune system or metabolism to affect Alzheimer's disease. And so if we can understand how all of these factors play into this system, we might be able to change things to really shape risk versus resilience and find the factors that can promote healthy brain aging and resilience to all of these potential risk factors. So first, we wanted to see, the field wanted to see if the gut microbiota could contribute to the signs of Alzheimer's in mouse models. And so a really fantastic lab, Sam Sisodia's lab, first asked this question by giving antibiotics to mice. What they found is when they wiped out the gut microbiota with antibiotics, there was lower amyloid plaques. There's less, you can see there's less green dots here. But the fascinating thing is that this only was protective in males, not in females. And this has been consistently reproduced in the Sisodia laboratory over a course of maybe 4 or 5 different papers. And I think this brings us to a really important point that that sex matters, that these interactions are different in men and women. And I want to be able to design, the microbiome intervention strategy for my mom and for my dad, and I want them both to work equally well. And so it's important to think about how these factors interact. One thing that you might not know is microbes can actually change your hormones, and your hormones can change your microbes. And, just some anecdotal stuff. Some people know that, if a woman is prescribed oral contraceptive, she goes on antibiotics, she's told that they're not effective anymore. And that's because microbes control the levels of hormonal reuptake in the gut. What happens is we excrete hormones in the gut. They're tagged to leave our system, but microbes can cleave that exit tag off of them. If they make this little enzymatic cleavage here, then the hormone goes back into the bloodstream and you have elevated levels of hormones. And so by changing microbial populations, you can actually have different levels of estrogens and testosterone in your system. Many studies know that different neurologic diseases appear at different ages, and many are different across men and women. Women have a higher prevalence of Alzheimer's disease, and we can think about the levels of hormones throughout the aging process, in which in men, there's a rise in testosterone during puberty, and then there's sort of a slow decline, but a slow and mild decline of testosterone over time. Whereas in women, again, there's a rise in puberty. It cycles during midlife, and then following menopause, there's actually a fairly rapid and marked decline over time, which may be related to changes in the gut microbiota and also Alzheimer's risk. And I think that's a really important area for us to expand our studies. Another factor that we really are interested in is aging. And what we see is the gut microbiota can change over time. It increases in diversity levels. It becomes very stable in mid-adulthood and throughout midlife. And then in aging, with immunosenescence, as we're unable to control our microbes or fight infections as well, there's a loss in a stability. Studies so far have actually shown that the aged microbiome can contribute to increased inflammation, reduced ability to fight infection, and also can reduce neurogenesis. And I think the really exciting thing as well is studies have shown that this is partially reversible. And if you give back young microbes, you can have a rejuvenating effect on both the immune system and on neurogenesis. So I'm really excited. I think there's a pathway forward in which we can really discover what are the young microbes, what are the microbes to keep our brains young. Another question that we wanted to specifically ask is how can we control this aging microbiota to promote a healthy brain? And so for this, we turn to looking at changes in the diet. Studies have shown that a calorie restriction diet, where you cut out calories from your diet, you're always at a calorie deficit, can increase longevity and potentially have beneficial effects in Alzheimer's disease. And so initially in these studies, my colleague Marissa Schaefer applied a 30% reduction in calories from carbohydrates. She specifically chose this. She did not want to have malnutrition. You don't want to lose fat. You don't want to lose protein. So this was a 30% reduction in carbohydrate calories. And the fascinating thing that she found is there was a reduction in plaques, but only in females. And so once again, we're seeing the sex-specific effect. In this case, the females are getting the protection. Then we looked at changes in the gut microbiota, and we really found there was this massive increase in Bacteroides in aging. The black line is our control animals. There's an increase, a doubling of Bacteroides in aging. This is when the mice are 15 months old, so that sort of corresponds to about 60 or 70 in these animals. And what we see is in our Alzheimer's mice, there's a tripling of Bacteroides in aging. And when we give this calorie restriction diet, we're able to keep those levels normal and equivalent to the young animals. Similarly, we saw about a 10-fold loss of a bacteria called Faecalibaculum that was lost in our Alzheimer's animal, the red group here. And starting this calorie restriction diet at 3 months of age in these animals, so about equivalent to their 20s or 30s, we were able to boost and maintain these levels over time. So doing this long-term dietary intervention was really able to block the plaques and block microbiota aging metrics. The one thing that I'll note, though, is that there are major concerns with malnutrition in aging. There's muscle loss, and a lot of people don't necessarily want to be hangry the rest of their life. So we're really looking for alternate strategies to promote healthy brain aging, tackling the microbiome without necessarily doing calorie restriction. And so through these studies, we identified Bacteroides as a target that might be linked to plaques. And so in our mice, again, we saw that elevation in Bacteroides. And in aging humans, in fact, we see an elevation in Bacteroides over time. In a study of individuals that were over 85, they looked at their gut microbiota and looked at health outcomes 4 years later. And the people with the highest levels of Bacteroides actually had an increased risk for mortality, showing that this elevation in Bacteroides over the usual levels could maybe have a detrimental effect. And in fact, in Alzheimer's patients, there's also an elevation in Bacteroides. So in order to really see if this bacteria could be contributing to plaques, we needed to test this in an experimental system. And so we administered Bacteroides to a model of Alzheimer's disease, and then we looked at plaque formation. And again, this is the very first start of the process in which the amyloid precursor protein is cleaved into these small peptides that form the A beta plaques. And indeed, what we found is you see the increased bright pink color here, we saw increased plaques when we gave this bacteria. We also wanted to know exactly how this was happening. And we've found that one of the major ways this occurs is by shaping immune responses in the brain. What we found in another experiment is if we administered Bacteroides and we looked at how well the immune system could clear the A beta, we saw this reduced clearance. And this is really teaching us that Bacteroides is blocking a crucial mechanism for immune-mediated plaque clearance, which is also depicted in the schematic here. And so if we can find ways to reverse this and actually increase microglia clearance of plaques, we can think about a way to restore and protect our brains long-term from the very early stages of AD pathology. Now we're going to go forward and also look at other ways to intervene besides just delivering bacteria. We've found in our studies that dietary interventions such as calorie restriction can prevent age-related changes in the gut microbiome. That happens differently in males and females, where females are protected. But we're doing new studies as well, and maybe a diet that's easier to do than calorie restriction. And we'll now test whether the Mediterranean diet can slow microbiome aging and maybe contribute to resilience against Alzheimer's disease. These studies really teach us again that the immune system can be at the heart of these interactions. So our gut microbes can change cells that line our guts that travel up to the brain, and they can change cells either to promote this stage where they can help you clear plaques, or they can lead to this immunosenescence that basically the immune cells will not do their job, they won't clear the plaques, and then damage can occur without a responsive immune system. And both in our studies, we're primarily seeing that the microbiome is driving this aspect of immunosenescence. It's shutting down our normal repair mechanisms. But we're also learning from the field that acute infections can drive inflammation, and that can also lead to problems in the brain. We've seen this a lot with COVID patients, that potentially people with underlying risk factors can have a COVID infection and then later develop signs of dementia and memory loss. And this could be, again, from an acute infection leading to very, very strong inflammation. And both of these pathways might be bad for brain health, and again, mediated by microbial agents. We'd like to think about therapy. We're learning these things so that one day we can intervene. We can think about ways to limit the bad microbes. So of course we can give antibiotics, which will hit a number of different bacteria, but there's new exciting approaches on the forefront, including using phage-based therapy, where we're looking at viruses or little chemicals called bacteriocins that are secreted by other bacteria that are really narrow in target. What these would allow us to do is really hit the specific bugs we want to decrease. There's also modulatory microbiome therapy. And this you can think about is eating your fiber, prebiotics, which are dietary compounds that promote the growth of beneficial microorganisms. Another area that's being pioneered by Howard Weiner at our center is to look at microRNAs that are produced in the gut, and these can selectively enhance the growth of specific bacteria. So again, it can be a much more targeted rather than a general approach. And then finally, you can have additive microbiome therapy. And in this, probiotics or live microorganisms are given that would confer a health benefit. We can think about fecal microbiota transplant, which is transferring the whole community, or what's known as postbiotics or microbial metabolites that can be synthesized into a therapeutic drug. And so I'm quite optimistic of the future of microbiome research in AD and related dementias, but I still think there's quite a lot of work that we need to do. So some of the major goals for me as a scientist and things that Cure All's Foundation is really helping us lead the way in is to help identify which microbes are actually contributing to or protecting against AD and related dementias. And this could lead us into new diagnostic screening methods. So everybody could go, at their checkup, check out their gut and see, do I want to change my gut so I can promote my brain health? We really want to understand how lifestyle, gender, biologic sex, race, socioeconomic factors can modify the microbiome and contribute to AD risk. And I think this will really help us expand our findings to the broadest community possible. We really want to find shared versus unique mechanisms across Alzheimer's, but also related to other dementias, also other memory disorders, so that we can understand when do we need a disease or Alzheimer's specific therapeutic, or when can I give something for a general dementia, maybe FTD or Parkinson's disease dementia. And then finally, I think it's really critical to consider developing guidelines for safe and effective microbiome-based treatments. Certainly, we want to make sure anytime you administer a microbe that it's safe for any population, and that we could test efficacy. And some of the things that I think are really going to speed up and accelerate this aspect is the use of new imaging technologies to see amyloid in the brain. This wasn't possible until, somewhat recently. We're getting new biomarkers every day. And so finally, we can see in our trials, when we intervene, can we actually move the needle? And I think that's really going to expand our possibility to develop these new therapies. And so with that, I'd like to say many, many people are involved in these studies. So I'd like to acknowledge some of the individuals, and especially my mentor, Howard Weiner, my laboratory, newly established Cox Lab. And I especially want to thank Cure Alzheimer's Fund for all the support they've given over the years that really helped us initiate and pioneer these studies. And thank you all for your attention.